Here we go. Hello. Welcome, everyone. I think actually this is my first call for this cohort. I just realized that I've been talking to you and I haven't introduced myself. So, hello, everyone. I am Sarah. I am the training and community lead for OLS. I work side to side with Gracieli, whom you've met already. And today in the call, we also have Precious helping with facilitation and hosting the call. And we have Laura Ondari, who's our expert for today. I'm just going to go through some housekeeping words like in every call. Some of them might be familiar, but if not, please, if you have any doubt, just drop us a message on the chat and we'll help you. So I'm encouraging everyone to introduce themselves from line 15 on the Framapad. Framapad is there pinned in the chat, in the Zoom chat. Don't forget to please add a W if you prefer to participate in the breakout rooms by writing, or with an S if you prefer just discussing out loud with the rest of your peers. Yeah, we're very curious about what TV shows or movies are tickling your fancy these days. Oh, I love The Good Doctor. I just saw that one. Amazing. Yes, we'll share the link again. So I just wanted to remind you once more that call is being recorded and transcribed. You can turn off your webcam now if you mind this. If not, or at least the breakout rooms, I remind you that they won't be recorded, so you can always turn on the cameras again so you can see each other. Captions are available. You just need to click the three dots in more and then go to CC show captions. That is exactly at least what my Zoom is showing. I want to remind you all that we work under the Code of Conduct and Community Participation Guidelines from OLS, which are linked in the Framapad. In general, we encourage everyone to participate as much as they can, as much as they want. We want to have a community that it serves all. That's why you are also all invited to our Slack workspace. That is the main interconnection and exchange channel for our community. Please do use it. Be considerate and respectful. We do love challenging discussions, but let's keep it always respectful. If you do experience or witness unacceptable behavior or have any other concerns, don't hesitate in contacting either Gracieli or myself. I think I just went a bit through the Framapad etiquette. Hi, Marjan. Yes, nice to see you. Yes, so we will be using the Framapad as a shared source of truth, let's say, and comments. Even if you discuss in the breakout rooms using the Zoom chat, which is totally fine, please don't forget to report out in the Framapad, because that's what Precious, for example, when we are discussing the outcomes of the breakout rooms, she can then check what you've reported on the Framapad. So even if you use the Zoom chat, which is totally fine, don't forget to at least have a small summary in the Framapad. And I think that's it for today. Oh yes, what I'm going to do as well is remind you, we do have, apart from these training calls, we have available coaching sessions for you with all of our experts. We have several topics. I'm just going to share my screen quickly so I can show you the sign up, which is very easy. So I think you should be all seeing my screen. Is that correct? So in the screen, you can see the Framapad. So if you click in the form that it is there, it will pop to a Google form. So these are all of the coaches that are available for you, the curriculum topics, but also other topics in which they are experts and they will be very happy to talk to you about. And you can just choose the sessions, the dates that you want to talk to them. You just need to add your name, your email, so you get the calendar invites. And very important, what you need to do as well is fill in this template, okay, this coaching session document. When you click in there, it will it will force you to make a copy. This is so you can edit your own template. So you make a copy, and it's open right now. And this is what you will see. Basically, this will be your own document for your coaching sessions. What we encourage you to do is please answer this set of 7 questions. Basically, so your coach can know you a bit better before or ahead of the meeting. Once you've done that, you just get this Google Doc link, put it in the coaching form, and submit. And with that, we will send you the calendar invite for the sessions that you've selected. You can select, I think it's up to 3 sessions, but if you need more, please contact us. And it can be with different coaches. The coaching sessions are not compulsory, but I would say please give them a try. They are super useful. They are very informal. You can also use them as out of office hours support sessions. Bring your concerns, whatever you're learning about open science. Yeah, they are usually very informal. You will be with your peers and with the coach, and having an hour just for you. So yeah, I do encourage you all to use them. And I think with that, I am just going to hand over to you, Laura. You have there in the Framapad again some space to make notes if you all want, or write the questions for her. And yeah, we will be using the Framapad for the activities afterwards. So thank you all. Thank you, Laura. Welcome, everyone.
Thank you. Thank you, Sarah, for the invitation to speak on this open data session. Sorry, my camera is down here on my phone and laptop, but I'm using my screen to see the slides. And Precious will be sharing the slides so that because I've had challenges trying to share them myself. So Precious, you can put them. So hello everyone, my name is Nyasita Ondari. I work as a bioinformatics research associate at the International Institute of Tropical Agriculture. I'm part of the communications and engagement team as well for Bioconductor. I am an open source manager at OSPO now under the Data Science Without Borders project. I'm also a co-founder of the Bioinformatics Hub of Kenya initiative, and in my free time, I am a podcaster. And I have a podcast called the Science in Real Life podcast, which I have not updated in a while, but I will get back to that. So today we are going to talk about open data. Most of us work with data every day, whether that's survey data, laboratory measurements, genomic data, images, computational outputs. However, generating data is only one part of the story. Increasingly, funders, journals, research communities, many people are asking us to think about how data can be shared, reused, and preserved for the future. So today we'll explore what open data means, why it matters, and how we can apply open data practices to our own work. By the end of this session, I hope you'll be able to define what open data is, understand how to find and use open data sets, learn how to prepare your own data for sharing, understand some of the challenges and limitations around sharing data, and become familiar with the FAIR principles. So I'm planning for this session to be as interactive as possible. So in case you feel stuck anywhere, please feel free to stop me, contribute your examples, and ask questions along the way. So before, Precious, we're now here. Thank you. Before we talk about open data, let's start with something more basic. What exactly is data? Data is any type of information that is collected, observed, generated, or created in the context of research. So for example, if you ask many people, they will think that data is spreadsheets. And for me, particularly, I will think data is genomic data sets, so FASTQs, FASTA files, and stuff like that, because that is the kind of data I work with every day. But data comes in very many forms. It could be survey responses, lab measurements, images, genomic sequences, and many other forms. So essentially, data helps us answer our research question. And not to be confused, data and information as well as knowledge are different, and that is what I was trying to show with this image on the right. So data is the atomic form of information. When you add context to data, it then becomes knowledge. And when that information is used to answer a specific question, then that becomes knowledge. We will not talk about wisdom because that is outside the scope of this presentation today. Next slide, Precious. So there are many different kinds of data, and the first type of data we'll discuss is primary data. Primary data is data that researchers collect and generate directly themselves. It's usually collected specifically to answer a particular research question. Because it comes directly from the source, we often refer to it as raw data. This is usually the starting point before any processing, cleaning, or analysis has happened. One common example of primary data is survey data. If you create a questionnaire and collect responses from participants, these responses are primary data. The information comes directly from participants and is collected specifically for your study objectives. Next slide. Another example is recorded measurements. So this could be, think about any measurements that you do in the lab. It could be blood pressure measurements for a clinical study. It could be crop yields if you're working with agricultural data like me. It could be rainfall measurements if you're working in environmental studies, or even lab instrument outputs if you're mostly doing molecular biology work. So these measurements represent observations collected directly from phenomena you're studying. Primary data can also come from biological specimens, for example, tissue samples, blood samples, microbial cultures, or even plant samples. Anything that you can use to generate that data in a lab can also be termed as primary data. Next slide. But not all primary data comes from physical experiments. So researchers often generate data from mathematical models and simulation. Increasingly, we've seen a lot of synthetic data, especially in my field. I don't know if it's the same in your fields as well. And that data can be used for example, climate models, epidemiological simulations, computational biology simulations. And you can generate, from these simulations, you can generate entirely new data sets. Although this data is computer generated, they are still considered primary because they were created specifically for the research question being investigated. Next slide. Okay, so there's also secondary data. Secondary data is data that is used by someone other than the person who originally created it. So this is extremely common in modern research. Many of us regularly download data sets from repositories and use them for our own studies. Secondary data can also be processed, it's cleaned and transformed to make it easier for other people to use. So for example, many share, for example, in my field, I'm going to use me as an example. I have used a lot of assembled genomes from public repositories to compare with mine. So rather than using the raw sequencing data that was obtained from sequencing platforms, I have used something that's already been cleaned, transformed, and preprocessed, and I have used that for my studies. That is secondary data. Next slide. We also have published data, and published data is data sets that have been prepared and shared for broader use. So I'm sure many of you have read publications and have come across supplementary data. And this data can either be primary, so it can be just raw, how the researcher collected it and deposited it there, or it can be cleaned and preprocessed for further use. The important difference here between that is that published data is usually documented, it's organized, it's made available in a way that others can access and understand it. Good published data is usually easy to find, easy to interpret, and easy to reuse. Next slide. There's also metadata, which is also a type of data, and this is often described as data about data. So think about a photograph. The image itself is data, but information about that image. So for example, when the photo was taken, where it was taken, which device was used to take that photo, and the file format are all metadata. So for example, here on the right, I've shown you like examples of what can be termed as metadata for particular data. And without metadata, data quickly loses its meaning. So in research, metadata provides the context that we need, and this context is needed by other people to understand and use our data sets correctly. Next slide. So what is open data? Now that we've discussed about data itself, what is open data? The Open Knowledge Foundation defines data as data that can be freely used, reused, and redistributed by anyone. The only conditions that may apply are requirements such as attribution and sharing under similar terms. That is what they call share alike here. The key idea here is reuse. Open data is not just about making data available, it's about making it possible for others to meaningfully use that data. Next slide. So when researchers are looking at data, they generally want data that is comprehensive, easy to obtain, easy to manipulate, and is believable. In other words, researchers are looking for data that they can trust and work with efficiently. So in the next few slides, we will see how to make data useful in that way. So first thing is that data should be sufficiently described with appropriate metadata. Remember that metadata provides context. Without context, it's difficult to understand how data was generated, what variables mean, how that data should be interpreted. So good metadata is what often separates reusable data from unusable data. The second thing is that the dataset should clearly explain licensing, copyright, and citation requirements. Users need to know what they are allowed to do with the data. Can they modify it? Can they redistribute it? Can they use it commercially? These are some things that creators should be able to put there in the data, and they should also explain how they would like that data set to be cited. Right. Yeah, third is that datasets should include clear access information. So users should immediately understand whether the data is openly available. If I can just go to a repository and download it, is it available upon request? There are some datasets that have that particular restriction. They also need to know what are the required ethics approvals. Do they require the ethics so that they can be able to access that data? Or is it restricted in any way? There are some data sets that are only available for certain research committees and not for everyone. So this access information should be tied to the data sets that people are using. Data should also be deposited in a trustworthy and recognizable repositories. So repositories provide long-term storage. It's easy to find them. They improve discoverability and often apply quality standards. So different repositories have their own quality metrics. A data set that is stored in a recognized repository is generally way, way easier to find, and people will trust it more than if you just opened a web page and put your data sets there. Right. Another important aspect is version control. So data sets often evolve over time. Errors may be corrected, new samples may be added, or additional processing may be performed. So for example, in my field, the first set of the human genome that we were using 10 years ago is not the same one that we're using now. Researchers have gone back and generated different versions of this human genome. And versioning allows users to know exactly which version of the data set was used in an analysis. If I was going to do an analysis today, it will show that I used this version that is most recent as opposed to the version that was used many years ago. Next slide. Yeah. So finally, data sets should include information about any processing steps that have been applied. This helps ensure transparency and reproducibility. If data was filtered, for example, if it was normalized, if it was transformed in any way using any statistical methods, the users should be able to understand what happened to that data. So these are some of the key things that researchers are looking for when looking at data. So benefits of data, open data provides many benefits. It supports reproducibility because others can verify your results. It promotes transparency because the evidence behind conclusions is visible and people can see and replicate that. It also enables validation. If other people can validate your results, they can reuse your data. They can then eventually become new collaborators in the work that you're doing. So many important discoveries, of course, up till now have been made using data that was originally collected and completely used for different purposes, but now is being reused for other reasons. Next slide. Open data also benefits the society. It can support policy decisions. It can help governments respond to public health emergencies. Think about COVID-19 between 2020 and 2021, because people were openly sharing data and also like their findings. It's enabled other people to be able to find vaccines much quickly, as opposed to if everyone was doing their own proprietary and closed ways of working. It also enables citizen science initiatives, and it helps ensure that knowledge is more widely distributed rather than being locked behind institutional boundaries. Next slide. And beyond that, researchers can also benefit directly. So open shared datasets can be cited, increasing visibility and recognition. Sharing data can create opportunities for collaboration. Of course, I have cited the public health emergency back in 2020. It also provides long-term preservation, ensuring that your work remains accessible, even if you move institutions or change projects, which is something that often occurs with us researchers in different fields. Yes, so I'd like to take a pause here for a moment. So think about a time when you needed access to data from another researcher. Was it easy to obtain? Was it well documented? Or perhaps think about a time when someone requested your data. What were the challenges that you encountered? So I will ask for people to put in their answers in their file pad, and then we can go through that. This can just be like a 2 minutes, 3 minutes breakout session, not breakout, reflection session.
Do you want breakout rooms?
No.
Okay.
No, this one I just want people to put it on the front pad. I don't know. That should be line 120. Yeah.
Just remove.
Okay, I can see some activity on the front pad. So someone has said we typically organize data sharing as a major part of our work, and so are bound by many regulations which we need to navigate to ensure our processes comply. Yeah, that happens in a lot of research institutions and even just funder regulations that can happen. Someone else said data sharing can be anxiety inducing as you can worry about errors being found. However, this encourages more quality control. So true, because if you don't share it, then maybe you will not be able to see the errors that you may have performed in your analysis. But then, I can understand the anxiety, but it's usually good to share when you can. Okay, I'll read a last one. Precious, do we have more on the chat? Maybe.
No, there's none on the chat, just mostly on the farmer pad.
Okay. There was a time in a previous research from my lab in which we needed to access an FPKM normalization script from a paper published in 2019. The researchers shared the script with us. after we exchange some email. Yeah, that also happens when you ask for access from them. They're usually able to provide that. I like that most of us have had an encounter with sharing data or asking for data from other research groups for our different research needs. It is really good. Do we move on to the next one, or do people still feel like they have more to share on the pharmacology?
Some are still typing. I think someone was working on a comparative genomic analysis. This is something you like.
Yeah, I was recently working on a comparative genomic analysis for some particular underrepresented lineages. It was such a huge relief to find documented genomic data on certain repositories. It helped a lot. Data sharing enables reproducibility and building on an existing knowledge. I couldn't agree more. It really helps that with building on existing knowledge. And yeah, I'm so glad you found well-documented genomic data sets. Sometimes it's usually difficult for that to find that, but I'm glad that was your experience. Okay, and the last one, data sharing helps us increase visibility of works and to enhance transparency of the works, enables us to advocate for open science as an exemplary. Yeah, true. I agree with that as well. Okay, precious. Good. Thank you so much for engaging with that prompt and question and rather activity. That was good. Okay, so at this point, it's also important to make it clear that open data does not mean that every data set should be shared publicly. There are situations where sharing data can cause harm. So before sharing data, we need to think carefully and ask ourselves who might be affected and whether there are any risks that can be associated with making that data available. This is usually, especially for sensitive data, which we will see in the next slide. So some categories of data, in this case, sensitive data, require special consideration. The first type of sensitive data that we're going to look at here is personal data. So this is information that can identify an individual directly, either directly or indirectly. So think about names, phone numbers, email addresses, national ID numbers, passport numbers. These are things that if anyone sees a phone number, it's usually directly tied to a person, right? And then there are also indirect identifiers, which may seem harmless on their own, but when combined can identify someone. So for example, age, sex, occupation, location, if you put them together, they may be enough to figure out where or who a person is. So this is especially important in health research because we often work with human participants. It's important to categorize personal data as sensitive data that should not just be shared willy-nilly. The second category is confidential data. So this is information that can be shared, that has been shared with expectation that it will remain private. So for example, confidentiality agreements, NDAs, government agencies that have internal records and other research for internal records and other research purposes. So even if this information will not identify people, it may still be under legal and contractual obligations that will prevent that from being shared openly. And also, if you combine personal data and confidential data, this would also be categorized as sensitive data. And the fourth category is biological data, which at first glance will always seem like it's not sensitive. But think about, for example, human genomic data that is unique to an individual, and it can be narrowed down to family members. And this can also reveal ancestry, disease risk, familial relationships. So because this genomic data needs some level of control, it should not be fully open. And then the final category is metadata. We have seen that metadata is data about data, and sometimes it may seem harmless, but think about, for example, GPS coordinates of endangered species. This could enable poaching if people find it, because not everyone on the internet or on open repositories has good intentions. Some people are malicious. Also, location information about, for example, archaeological sites could lead to looting. So this metadata can, help people trace back to the original data. So when we think about sharing data, we need to think carefully about the metadata as well. Next slide. So not all sensitive data carries the same level of risk. Some data is considered highly sensitive, and these are obviously things like patient medical data, bank details, salary information. This should not be information that's just shared willy-nilly. If that happens, it will cause serious harm to both individuals and organizations. So think about the person themselves, or, for example, the hospital from which this patient data was leaked. There's also medium sensitivity data, and this data includes things like contact information or student records. While it's not as damaging as highly sensitive data, it requires protection and controlled access. So not everyone should be able to just go in there and find it. And there's now low sensitive data, and this is usually information that's not intended for public release, but would cause a major harm if it's accessed by others. So one thing we always need to know is classify our data and start thinking about how we are going to share it. If we understand the level of sensitivity that our data requires, that will enable us to look at ways to share it without being inappropriate or causing harm or risk. Next. Yeah. So to summarize this section, there are several situations where data should not be openly shared. The first is when that data contains information that could threaten national security. The second is when it contains personal or medical information that could compromise privacy. The third is when there are cultural or indigenous data considerations. And the last one is when there are intellectual property concerns, especially if the research has commercial applications. Open science is not about sharing everything. Usually there's a phrase that says it's as open as possible, but as closed as necessary. Yeah, and so this will lead us to our first breakout session. So for the next few minutes in your breakout rooms now, think about what are the biggest barriers that prevent researchers, especially in your fields, from sharing data openly. And it would be good if you share some personal experiences for that. Sorry, this next activity came too close to the previous one.
It's okay. So I am assigning breakout rooms now.
Yeah. Mhm.
Let me see. We are about 1, 2, 3, 4, 5, 6, 7, 9. Crazy. We're about 18, so I think we can do maybe 6 people. Is that so much?
Yeah, that's good.
Okay, so 6 people per
You have your breakout
room.
room? Yeah.
Yeah, okay.
We
So,
can do this for about 10 minutes or so,
alright, that's
and then
okay.
we'll go through. Yeah.
So, okay. Laura, would you be in the breakout room or you be in the main room? You want
No,
to join
no,
one? Okay. So,
I would not like to join.
alright, cool. Um, hey, okay. So, let me see. Um, okay. Mm, I think everyone should most likely have one now. Mm, okay. Some have dropped off, so I can't see. Sorry. Okay. Let me know if you don't have a breakout room yet. I think I should have assigned almost everyone.
I'm not in breakout room.
Okay. So you should get like a notification at the top of your screen. Oh, which is a new. I think you're the only one left. I will assign you to a breakout room. Sorry, Natalie. Let me fix that. That's my bad. I'm sorry. Mm. Sweet, oh, yeah. Okay. All right.
Precious, are you going to be in a breakout room yourself?
I will just float around to encourage people to um, write on the chat. What are the Hi GT, welcome. Let me know. Please write on the farmer pad if you want to be in a written or a spoken breakout room, and then I will assign you as soon as possible.
Precious? Precious? Precious?
Sorry, I was muted. Can you hear me? Oh no.
Could you hear me?
Yeah, I can. Can you hear me?
Yeah, I want to. My phones are dying, so I'm trying to go back to my
That's
system.
okay. That's very fine. Hi Victor, welcome. Please let me know on the please add to the camera pad what if you want to be in a written or in a spoken breakout room, and I will assign you.
First spoken room.
Okay. Let me add you to one. Good. Hi, Aline. Welcome. Were you on the call before?
Hi, Precious.
Hello, are you back?
Something wrong somewhere.
We're back.
Okay. Thank you so much to everyone for participating in the breakout room. So for the next like 3 minutes, maybe we'll take turns and just share what our biggest barriers and worries are about sharing our data. And for now, I'll not be reading them from the Frama pad, so please feel free to unmute and share with the rest of the group. We'll start with someone from Breakout Room 3. Anyone from that breakout room? I can see Claire, Michael, Zainab, Mohammed, and Masume were in that room. So could anyone share with us what you guys discussed about?
Yeah, sorry, there was somebody still typing and in the notes there. So
Okay.
we basically noted that there may be a variety of barriers and worries around data sharing. So it could be around identifiability. It could be that people are concerned about criticism if they've made mistakes in their data or their interpretations. There may be IP worries as well by sharing too much openly. They may fear being personally held accountable if there's something accidentally shared that they shouldn't have access to or shouldn't be sharing. There could be, sorry, I'm just looking at the last couple of notes that were added on. They could be worried about people not crediting them correctly with the appropriate license and that reducing their competitive advantage. And also there could be a lack of appropriate infrastructure and tools and a lack of open science knowledge within their own institutional culture.
Oh, excellent points. Thank you so much. And I like that you came up with several different scenarios for that in your breakout room. Okay. Could we have someone from breakout room 6? And you can share some that Claire has not already mentioned so that, we add onto the barriers and fears. Is there anyone, or should I just name someone?
Okay, in that case, maybe there's no one from Breakout Room 6. Anyone from Breakout Room 7 that would like to share?
Yeah, I can
Okay,
go.
okay, all right, so you can go ahead, Adam.
Sure. We said that researchers would worry about sharing sensitive data, so perhaps by accident. Epuka also said that there's a barrier that other researchers could claim the findings or the ideas from the open data. There's a barrier is knowing how to do it correctly and where to share it. Another barrier is that it's not rewarded or recognized and takes time to learn that could be spent on other things. And similar to might have already been said, the idea around kind of intellectual property and commercialization concerns as well. They might want exclusive rights over their information. Thanks.
We're also going to, thank you so much for that, Adam. We're also going to be looking at some of the ways to share in subsequent slides. So I'm glad that that came up so that we can see how to tackle that. Thank you. And then the last room would be room 1, which had both written and spoken participants. So anyone willing to share? I can see that you all are still typing, but you can just share 1 or 2 points and then we can move on.
Okay, can you listen to me? Okay, so my name is Charlie. I was in group 1 with Anu and Owasinke, and we tried to answer the question from the perspective of our own work, from our researches. And most of us work with clinical data or data collected from humans, personal data. And we consider that sharing this kind of information has a lot of limitations regarding some legal regulations that prevent us from sharing some aspects of our work that are ethical aspects that we need to consider. We had a great discussion, for example, that sometimes sharing the data that we collect could be a great way to increase awareness of our work. It could be a great way to attract, for example, grants from people that catch some interest in the research that we're doing. But at the end of the day, we need to consider that this is sensitive information. As you talked in your presentation, there are some levels of sensitivity that we need to consider. And even though sharing is great regarding the awareness of the research, we also see that there are a lot of balances that we need to make, and just sharing everything could be detrimental to ourselves and even to the participants that kind of, trusted us.
Yeah.
So that's it for me.
Thank you so much. Yeah, all of you have given excellent points, and for sure these are because all of you are researchers. Okay, not all of you, but most of you, I guess. These are some of the dilemmas that we face every day when we're doing our research and sharing our data and how to make it open. So I'm glad that these are actually real research scenarios that have been brought to the discussion. And thank you everyone for participating in that. So now we will move on with the presentation, and hopefully I can make it through the next slides in 20 minutes so that we can have time for our last breakout session. Okay. So now that we've talked about sharing data, we're now going to switch perspectives and think about finding data. One of the most common places to find data sets is in repositories. There are general purpose repositories like Zenodo and Figshare and Mendeley Data that accept data sets from any discipline or many disciplines for that matter. But there are also domain-specific repositories that focus on particular fields. So for example, in bioinformatics, there are many of us use NCBI for sequence data, or for example, I use Protein Database for protein structures. And there's also many universities and organizations that maintain their institutional repositories and encourage their researchers to deposit data in those repositories. But there are also government ones like the data.gov for the US, people who stay in the US, and they provide national repositories through open data portals. So next slide. Our repositories are useful when you already know where to look, but search portals are useful when you don't. So tools like Google Dataset Search, Kaggle that has a lot of machine learning and AI datasets, Wikidata, or even Dataset Commons allow researchers to search across many datasets for different sources. So think of them as engines that are specifically designed for data. So you can go in there, log in, and find something when you don't know where to look. Next slide. Yes, so publications are another valuable source of data. Many journals now require authors to deposit supporting datasets in repositories and provide links for that within the publication. And also there are supplementary files that can contain useful data, although repositories are generally preferred because they provide much better long-term preservation of that data. So finding a data set is only the first step. So the next question is whether that data set is actually suitable for your research. Not every data set is openly available. Not every data set, that is already openly available is necessarily useful or reliable. So it's important to evaluate the data that we are seeing before we invest time using it. And when assessing a data set, we usually start with practical questions. So is that data well described? Can you understand why it was collected? Is it intended? Is its intended use clear? Is the data accessible? Is it current? And will it still be available in the future if you want to revisit to try and answer the same research questions later on? So these questions help determine whether the data set is likely to be useful and sustainable, and is a good assessing metric. Next slide. So the next thing is to think about technical considerations. So is that data available in the appropriate format? Can your software read it? Is it machine readable? Are the acronyms and technical terms used in that data set defined? And is there documentation explaining how to work with the data set? So for these technical considerations, these are factors that often determine how much effort was put or is going to be required before your analysis can begin. Next slide. Sorry. Finally, there are social questions. Who maintains that data set? Can someone be contacted if issues arise? Is there a community already using the data? And popular data sets often have active communities. So think about, for example, NCBI, that the community is active and usually people are versioning and changing that and can answer. They have contact information there. That usually provides support. And that is a very good example of good practice when you're assessing data to use. Next slide. So when we're using data or when we're using someone else's data, we should acknowledge their contribution. Just as we cite publications, we should also cite data sets. Data collection often requires years of work and really substantial resources and infrastructure from institutions and individuals. So proper citation ensures that data creators receive credit, and it helps others locate the exact data set that was used. And this is just an example of a citation here that I have included. Just the same way you cite a publication, you'd also cite a data set, and whether you've gotten it from a data repository or a search portal or a publication, more often than not, it would use this same citing criteria. Next slide. So now let's focus on preparing our own data for sharing. One of the best practices when starting a project is creating a data management plan, often called a DMP. So a DMP helps us think about how data will be collected, stored, managed, preserved, and shared through the project lifecycle. So a data management plan should answer several important questions. So what data will be collected? When will it be shared? where will it be shared, how will others be able to reuse it, and who is responsible for each aspect of this data management. Having these discussions should be had very early in the research cycle because they prevent problems that might be had later. Next slide. Yeah. So one important part about making data sharing is selecting appropriate formats. The goal is interoperability. In other words, we want other people to be able to access and use our data without unnecessary bias. So when choosing formats, ask yourself a few questions. Is the format compatible with your data? Does it support metadata? Are there tools available to read it? Is it commonly used in your field? Are there community standards that guide how you can use this data and how collaboration can be gotten from using this data and how reusing can be made much easier? Next slide. So these are examples of non-open formats that can create challenges for reusing data. So for example, if you're using Photoshop, and this .psd data formats will require Photoshop when you're trying to open that data. If you're using Word documents, they will require specific software to be able to open. I'm sure most of us may have come across when someone has provided supplementary materials and it's using Microsoft Word or Excel or whatever. Nowadays, you need to pay for Microsoft and it just says cannot open if you don't have Microsoft. Another example is AutoCAD files that require specialized design software to open them. So the problem is not that these data formats are bad. They're not bad. It's just that they limit accessibility. Next slide. So open formats are generally preferred for sharing. And here I have, in this table, I have shown a couple of formats that are preferred. So for example, CSV files, which are comma separated files, are simple and are widely supported. We also have HDF and CDF formats, which are useful for large and complex data sets. The ISA framework is commonly used in life science research. And usually the key advantage of this format is that they can be accessed by a wide range of tools and users. Next slide. So documentation is what allows someone who has never met you to understand your data set. So good documentation will always explain how data was collected, what variables mean, who collected that data, how was it collected, how is that data set cited if anyone reuses it, and what are the limitations that users should be aware of. Slide. So in addition to documentation, we should also provide version information and licensing information. Versioning helps users understand which release of the datasets they're using, and licenses explain what others are allowed to do with that data. There are different licenses, and I'm sure as part of this course that you're undergoing with Nebula, they will be explained in later. Either you've already been explained to or it will come later on in the sessions. But for example, there are usually CC licenses for datasets, and the CC0 allows you to reuse and does not require citation. But if you use CC BY, you have to provide that particular citation. Next slide. So at this stage, we've prepared our data. We've documented it. We've thought about formats and metadata. The next step is usually actually sharing that data. And sharing data is not a single action. It's usually a process. And there are several things that we need to think about before our data sets are ready to be shared. So the first question is whether that data should actually be shared. And have we met all the ethical requirements that are needed? Next slide. So have personal identifiers been removed? Do you have permission from collaborators and funders and participants? And then once we've established that the data can be shared, The next step is selecting an appropriate repository. The repository becomes the long-term home for our dataset. Ideally, it should be trusted, widely recognized within your field, and committed to long-term preservation. So, for example, sequence data might go to NCBI, while a general dataset could be deposited in the Node or Figshare. So repositories often have their own requirements. They may require specific metadata fields. They may also require particular file formats, naming conventions, or documentation standards. And this can always feel like a lot of extra work, but these requirements actually improve the quality and usability of a data set. So simply uploading a data set isn't enough. People need to be able to find it and access it. That means providing clear metadata, meaningful descriptions, keywords, and ensuring that access conditions are transparent. A data set that nobody can find might as well not exist. We also need to request a DOI. One of the most valuable things is usually this DOI because it's a permanent identifier that allows others to reliably locate and cite your data set. Just think of it as giving a permanent address to your data set. And then next slide. Sorry, back. So I can explain that last point. Yeah, choosing a data license. So the final step can be getting an appropriate license. We've already looked at that before. That license will tell users what they're allowed to do with your data, if they can reuse it, if they can modify it, and do they need to provide attribution. So without a license, people will not know how to properly acknowledge you for the data, and they may not legally know what to do with your data. Okay, next slide. Okay, a common question is usually, when should I share my data? There isn't usually one universal answer. The timing often depends on the project, the funder. It could be journal, your institution, the type of data involved. And for example, here, advanced sharing means making data available during collection or shortly afterwards. So some large international projects follow this model. The advantage is that it maximizes opportunities for reuse and collaboration. But the challenge is usually ensuring quality control before release. So a good example would be like if you have pathogen surveillance, where that sequence data is often shared very rapidly because there is immediate public health value if it is shared early. Next slide. There's also intermediate sharing, which occurs when a study is published. This is probably the model many of us are familiar with, especially in our research fields. The publication is usually released together with the data, and that allows for reproducibility. So many journals nowadays require that you publish your data as part of their publication policies. Next slide. Sorry. So there's also minimum sharing, which generally means making data available at the end of a grant or project. Even if the data has not yet been shared publicly during the project, the expectation is that scientifically useful data will eventually be made available. And then there are also cases where there's no sharing, like sharing is not possible at all. And this, as for example, the participant from room 1 had said, this may be due to privacy concerns. There are also legal restrictions, ethical obligations. For example, indigenous data as well. There are also governance considerations that should be looked into and intellectual property concerns. In this situation, the decision to not share may actually be the most reasonable decision. Next slide. So sharing considerations. So when selecting a repository, there are several practical questions we should ask ourselves. So sometimes funders require you to put your data in a particular repository. So ask yourself if your funder requires that. Does your institution have recommendations? Nowadays, different institutions, as I had mentioned before, require you to use their repositories to deposit your data there. Also, is there community repositories that researchers in your field already use? It would be good to put your data there for discoverability and for others to find it and reuse it, or maybe ask questions about it. Also, will the repository remain available in future? That's a really good question, especially if you want to revisit it or if other people in your field want to contribute to that knowledge. It would be good if that repository will still be sustainable. And of course, importantly, does it support FAIR principles, which is findability, accessibility, interoperability, and reproducibility, I think. Next slide. Just next slide. Okay. But usually there are misconceptions when it comes to data sharing. Many researchers think that sharing data means emailing a file to someone or uploading it on a personal website. While technically that is sharing data, it does not provide long-term preservation or discoverability. So repositories are generally a much better option because they provide persistent identifiers, which we've talked about, the DOIs and stuff. Repositories also support metadata support, and they also allow for long-term storage. Next slide. Okay, so when depositing data, repositories may ask for sample files or perform checks before publications. This helps that data is complete, the metadata is correct, and that the data set meets community standards. So these are sort of the quality control processes that they will follow. Next slide. Okay, so data sharing doesn't necessarily end once the data set is published. Sometimes errors are discovered, sometimes new data becomes available, sometimes documentation needs updating. So repositories provide that, but for long-term preservation is really one of the main benefits of repositories. A good repository should continue maintaining access to the data even after the project has ended. This ensures that data sets remain useful even for future people. So this is really important when you think about archiving your data in a repository. Next slide. So to enable data reuse. So ultimately, the goal is not simply to store data. The goal is to enable reuse. So if you provide a DOI, you ensure findability, you supply good documentation, you make it easy for people to cite you, all this increase the likelihood that others will use your data. If someone finds your data, is able to understand it, can confidently use your data, then you've done a good job. Okay. Right, so this is actually one of my favorite slides because people often assume that data sharing is the responsibility of a single person. In reality, it takes a team. So the PI may oversee the project, and then researchers will generate the data, data managers will curate the data, bioinformaticians will process it. For example, here in our institutions, that's what we do. Then other people may be tasked with supporting deposition and preservation of that data in different repositories. Open data often works best when responsibilities are clearly defined from the beginning. So when you're designing your project, think about who will move that data to a repository, who will develop the documentation, who will be responsible for the data used, and who will guide the privacy concerns of that data. And that is one of the reasons why a data management plan is so important. And I'm sure this will be covered in the next part of open data. This was part 1, so I'm sure DMPs will be covered in part 2. And so now we get to our third activity, and this will be a breakout session. I hope we can be able to use the next 6 minutes for that. Yeah, so Precious, you could open maybe for 5 minutes. People can work on it and then we can come back and close. And this is just an example of what a research team conducted a study, and this is how they shared their data. So we've put this on the Pad on line 235. And then the discussion questions are from line 248.
Do you want to answer the question that we have before we move to breakout rooms?
Oh, do we have one? Sorry, I didn't
Yeah,
see.
it's okay.
Could you read
What's
it
the
out to me?
difference between
Mhm.
data replicability and, I'm sorry, yeah, sorry, data reusability and data reproducibility? Like, is there a difference between
Data.
reusability of data and reproducibility?
I think reusability is just someone taking that data and reusing it. They could apply different research methods, or rather processing methods to that data, by reusing it in a way that you intended for it to be reused. But reproducibility could be on like different environments, but coming up almost with similar results that you got when you were analyzing that data. So that replicability is the exact same methods with the exact same data. Reproducibility is that data with different research methods, but coming to almost similar results that you got.
Great. So I am opening all the rooms now. We might have to use this one for a while. But yeah. Okay. You should be able to join your rooms back. Yay. Amazing. Hi Esther, would you like to be assigned to a written or a spoken breakout room? Resume recording. Right.
Right. I don't know if that was enough time to walk through the activity, but I'm so sorry we've gone overboard by 5 minutes. Okay, or more. It's now 6 minutes, but we'll just go through the discussion questions really quickly. We'll not go through all the solutions for now. So I don't know how do we do this? Do we ask people, or that will take time? Yeah, call facilitate. Precious. Did everyone finish discussing the questions, or you guys would like more time to just keep looking through them? And you, during the cohort sessions, you can discuss amongst yourselves. Okay, those who think we should discuss now.
I don't know. For me, maybe I don't know if others have some time restrictions. I could maybe just share a little bit about my group.
Yeah.
So I'm Charlie from Group 1, and alongside Anu and Owasinke, we basically discussed that there are a couple of problems with this work. They did not publish a DOI for the data or DOI. They did not deposit the raw data or most of the data in public repositories, so this hampers the reusability of the data. We did not think that this data is particularly findable, even though they mentioned the data set on the paper. We can only access a spreadsheet, which even the name of the spreadsheet is not quite good for reusability in the future. So there are concerns regarding the format of the data. Maybe they could include licenses if they start uploading this data to a repository. They should consider actually including licenses so people can actually feel comfortable in using their data. And particularly for me, as I was talking to the group, it felt like they just had this, that they did not think of making this an open science work from the beginning. It wasn't designed to be an open science work. They tried to make it after the work was done. So they just started publishing things. But it is not enough to say that this is an actual reusable, findable, and an open science work. They could do they could tweak a little some things, but there are some concerns that should be actually resolved. That was it from my group, I guess. If others want to maybe add their own views, be nice.
Yeah. Yeah, I'm sorry. If anyone needs to leave, we're already past the mark, so you can. But I like the points that have been shared here. And yeah, so they named their dataset final_data_v3, and it was an Excel format. Yeah, that's not particularly findable. It is partially, but not really. So you don't even know what final data means. There's no particular description that makes that to be known what it is. Of course, there were also no metadata or keywords provided. There was no DOI as mentioned, and it was not deposited in a repository. That's true. It was also not accessible in a sustainable way, because while you can download the file now freely, there are still concerns because personal web pages can be removed. Sometimes you can have a .com and then you fail to pay for the domain in time, and then it goes to someone else. And yeah, it can become unavailable over time. There's also no guarantee for long-term preservation, no repository archiving, no information about licensing, as he mentioned. Yeah, and is it interoperable? Because it's an Excel file, an Excel spreadsheet, it's not necessarily open data format. And other researchers may not know what variables were recorded or classified. This data set can only partially be reused, but there's no data dictionary explaining variables. Again, no metadata, no licenses. Yeah, and no information about quality control or data cleaning, which is true. There are also concerns related to privacy and confidentiality. So there are exact hospital names, patient ages, hospital locations. Yeah, and although this may not be direct, but they're there, and that's a potential concern. Yeah, so thank you everyone for attending this session. I'm sorry we went over time, but I really enjoyed taking you through the slides and seeing everyone's contributions on the Frama part and during the call. Thanks so much.
Thank you. Just like reminder that we have the next cohort call on open tools and resources for June 16th. So that's 3 p.m. UTC. Then there are coaching sessions with the weekly deadlines to sign up. For sessions on Wednesday, please sign up by Sunday. And then for sessions on Friday, please sign up by Tuesday. If you have any other question for Laura or myself, you can always put it on Slack and we would follow up. The link to the slides are on the Frama pad. So thank you so much for today's session. See you all around.
Bye.